So it's my pleasure to introduce Ha. So Ha is a PhD student in the Bioinformatics program, finishing up her 5th year. And the work she's going to tell you about, it was on universal chromatin state annotation of the human genome. So this work, it was recently published this year, and it's something which already is being used in multiple projects, both in my lab and with various collaborators, and I think it might be sort of broadly useful. And I'll let Ha tell you more details about this work.
Hi, can you see my big screen or my smaller presenter view? Which one do you see?
Yeah, it's good now.
Okay. Yes. Hi, everyone. I'm really happy to share with you about my research project called Universal Annotations of the Human Genome through integrations of over 1,000 epigenomic datasets. Yes, like Jason said that this work has been published. I actually gave a presentation about this particular project at QCBio retreat in 2019. But since then, we had made the manuscript. We went through multiple rounds of revision, and it just got published earlier this year. So this presentation we will try to present more of the many progresses that we made to the now published manuscript. But first, let me give you a little bit of background about this particular project. So epigenomic marks can help us, epigenomic marks such as histone modification, histone variants, or DNA hypersensitive sites can help us reveal a lot about the dynamics and locations of DNA regulatory elements. And these epigenomic signals can be a powerful source of information for annotating the genome. This is just a screenshot of genome browser screenshots of a region of the genome where each track here correspond to one histone modification mark, or what we call a chromatin mark. And given that you can provide many different chromatin mark across the genome, we can use software such as CHROMHMM, which Jason developed, to learn the spatial and combinatorial patterns of the chromatin marks, and we will then be able to classify the regions of the genome into what we call chromatin state. So for this particular plot right here, you can see that these are the tracks of the signal of different chromatin marks, and this track right here show you the chromatin state annotations of each of the regions on the genome. The highlighted region right here are all annotated into a chromatin state that's color red, and it's actually a TSS promoter state. And you can see that these regions actually correspond to region with really high signals of these two, or jointly high signals of these two chromatin marks. And so yes, that's what CHROMHMM really tries to help us figure out in terms of genome annotations. And the states that we can learn from the CHROMHMM model can actually be characterized in terms of their biological implications through looking at the emissions matrix and the external enrichment with many external genome annotations, which I can go into details later. The CHROMHMM model actually has been used to produce chromatin state annotations for 127 different reference epigenome as part of roadmap and ENCODE projects. This is just a plot showing you a snapshot of the genome where each rows correspond to a reference epigenome and the columns correspond to different regions of the genome. And we can see the color actually correspond to different chromatin state that are used to annotate the genome of different reference epigenome. So what I just show you over here are actually the output of what we call the concatenated model of CHROMHMM. And this concatenated modeling actually give us what we call per cell type, or you can also call cell type specific annotations. The way that this model is learned is that the input data from different cell types are actually concatenated into one long genome. So the input data actually much longer than our actual genome. And we input this data into CHROMHMM, and what we get as output is genome annotations. But these annotations are per cell type annotations, meaning that each cell type would have their own set of annotations. But the set of states that are used to annotate the genome are actually the characteristic of the state are shared across the cell type, meaning the red state have the same biological meaning across all the cell type, etc. And this has been used, like I said, to annotate the genome in roadmap and ENCODE projects. There's another way to learn the CHROMHMM model that actually has been kind of under study before is what we call the stack modeling approach. In this stack modeling approach, the input data tracks correspond to a combinations of chromatin marks that has been provided in a particular cell type, and all these input data tracks are stacked as input as individual input tracks into CHROMHMM. So we put all these signal data tracks into CHROMHMM, and we get the output is also a genome annotations. But this genome annotations is shared across all the cell types, and that's what we call the universal annotations because it's shared across the cell type. What we get is each genomic position, we get a state annotations of that particular position that can be used to annotate the genome across all the cell types. And the large scale, so that's actually the focus of our project because we actually introduce a large scale applications of the stack modeling approach, which has previously been underexplored before. And I want to present to you that the full stack model the stack modeling approach that we presented has some complementary advantage to the existing per cell type annotations for several reasons. The first reason is that the annotations that we get from the stack modeling approach is universal, meaning it's shared across all the cell type, and therefore it can help us differentiating constitutive chromatin's activities from the more cell type specific regions. The second reason is that for many applications of the genome, if we want to study the chromatin state context of different regions of the genome, it may not be natural to pick one particular cell type to work with. The example of such cases would be when we want to study DNA sequence conservations or genetic variations. In such cases, the universal stack modeling cell type and the universal annotation can be a better alternative. Another reason is that the stack modeling can help us leverage a much larger amount of input data for genome annotation, therefore give us a more complex system of annotating the genome. However, even though the annotation is more complex, it can also simplify the task of learning genome annotation across different cell types. Because what we have right now is at each genomic position, we have only one state to annotate that position, as compared to when you have per cell type annotations and you have to look at the chromatin state across all the cell type at each genomic position. So with that, I like to introduce the full stack model, what we call the full stack model, because we actually use input data from more than 1,000 different experiments into chromatin, and the output of that, we would get what we call an emission matrix. And in this matrix, you can look at all the rows are actually corresponding to all the chromatin state. In this case, we train a model with 100 full stack state, and therefore we have 100 rows. We actually have more than 1,000 columns, and each of the column correspond to one experiment, as one experiment data set that we input into the model. And the heat map here actually show you for each of the state, the probability of observing an experiment signals at each state. So, and a lot of our work in this particular project actually is to understand this particular heat map and to derive the biological implications of the state, of each of the state. And to do that, the first thing that we try to look at is to look at the chromatin marks that are associated with each of the state. So what we do is that for each of the state, so for each of the rows, we look at the top 10 highly emitted experiments, and we color the experiment by the associated chromatin marks. And we highlight an example here where you can look at this particular state in the DNase group. This state show really high emission probability for this cluster of data set. And this data set are all data set that are used to characterize the DNase chromatin marks. And correspondingly, you can see that all the 10 highly emitted experiment associated with the state are all experiments that are used to profile the DNase mark. And this is just a 2D plot of the emission probability for this particular state, where you can see a spike of emission probability for all the experiments that are corresponding to the DNase mark. So this state, therefore, can be characterized as like a constitutive DNA state. We also further look into this state and see that this particular state actually showed really strong enrichment for CTCF elements that are observed in 6 different cell types that we have the data for. Therefore, this state is a constitutive DNase and CTCF element state. We also look at the importance of cell type specificity in the different state. And what we did is we also look at the top highly emitted experiments for each of the state, and we highlight the experiments by the associate cell type that the experiments were conducted in. So you can see that I'd like to I'd like to highlight this particular state, which we call the actually the enhancer A6 state, and it shows that the top highly emitted experiments are all associated with enhancer marks, and all these enhancer mark, all these highly emitted experiments for this particular state are all used to measure enhancer marks in the brain area. And therefore this state, we can kind of hypothesize that this is a brain specific enhancer state. And we actually can confirm that by looking at, again, at this particular state, and you can look at the input data set. Across all the input data set, you can see that in the brain cell type right here, these data tracks correspond to data that are profiled in the brain. And in the brain cell type, you can see a very high, distinctly high signals of the mark H3K27AC, which is an enhancer specific mark for the brain cell type that show signal in region that are classified as this state. So this is to show you that the full stack state that we produce can actually help us differentiate regions that are kind of constitutive from those that are showing more cell type specific region. And this is just I'm giving another example of how the full stack state that can show rather cell type specific patterns. So this state right here is what we call the HET9 state, and this state is what we after multiple analysis we actually characterize as a state that are heterochromatin state, but specifically active in the embryonic stem cell. And you can see that in this, you can see that this is the full stack state that are highlighted as the heterochromatin 9 state. And in this region of the genome, highlight all the cell type specific per cell type annotation that are in heterochromatin state, but are in quiescent in all the other cell type. Therefore, this is an example of embryonic stem cell specific heterochromatin state. We also, to understand the biological implications of the full stack state, we also look at the enrichment of the state with annotated genome elements, such as exon, transcription, start site, transcription, end site, etc. So these are 100 different state in our model, the rows, and the columns actually correspond to all those external annotations. So we have a coding region, we have exon, transcription, start site, etc. So we can have some example takeaway. Some example takeaway is that, for example, this gap artifact state number 1 right here is a state that we show to be highly enriched with assembly gap in HG19. It's actually overlap with 99.9% of all the assembly gap in HG19. And another example is these two states that are in the group of transcription stat site, TSS 1 and 2 state, show that are the two states that are most distinctly enriched with annotated transcription stat site. It shows more than 100-fold enrichment. And there are many other examples take away from this plot, and we actually summarize all these in our state characteristic accompanying with the paper. We also look at the state enrichment, neighborhood enrichment around transcription end site and transcription start site. And this particular, these two plots would help us highlight certain states that are highly enriched around the neighborhood of transcription start site and transcription end site. And it can also help us further characterize a state in terms of their upstream or downstream enrichment with TSS or TES. We also looked at the full stack state enrichment with PHASTCONS elements and repeats element. And for those of you who are not familiar with PHASTCON, these are just elements that are corresponding to conserved elements, conserved regions of the genome. And these are the top 10 states that are most enriched with PHASTCON elements. And these are the top 10 states that are most enriched with repeats element. And and you can see that the top 10 states most enriched with PHASTCONS elements are mostly promoter or transcriptional activity state. And those associated with repeats element are more associated with heterochromatins or quiescent state. But the common characteristic of these states is that they all associate with the mark H3K9AME3. And we also, within the repeats element, we also look at the state, at the enrichment of state with different classes of repeats element. And so I'm just highlighting some of the state that are the most enriched state with different classes of repeats element. And so some highlight takeaway from this is you can ask, why is it that some transcriptional state TSS1 and TSS2 show such high enrichment with low complexity state? And one reason that we can explain for this is that these regions transcription stat site actually show really high enrichment with CpG island. And so these regions are CG rich regions of the genome, and therefore they also can overlap with low complexity repeats. Or you can also look at and observe that an acetylation state is really highly enriched with simple repeats. And one reason that we showed in the manuscript, but I don't show the plot here, is that the reason is because this particular state actually shows really strong enrichment with regions that are like a repeated region of CA or TG nucleotides. Or a heterochromatin state show really strong enrichment with long tandem repeat, and the reason is because long repeats tends to be rich in heterochromatin region, which are regions that are packed into heterochromatins in the genome. And there's many other association with repeat element that we also address in our manuscript. We also looked at the full stack annotations enrichment with structural variants. And here I highlight the top 5 states that are enriched with either deletions or duplications. And you can see that most of these states are in quiescent or heterochromatin state, but they also show strong signals of the mark H3K9Me3 right here. And we can ask ourselves, why are structural variants actually more enriched with H3K9Me3 associated with state? And one of the reasons is that structural variants actually are variants that have really large effects, and therefore they have a negative selection forks in a more conserved and functional parts of the genome, and therefore we can see them are enriched in the less conserved part, which is more less conserved, less functional part, which is corresponding to quiescent and heterochromatin state. And at the same time, our findings actually is consistent with previous finding that's found out that heterochromatin state and zinc finger state actually are observed to be enriched in structural variants. We also looked at the full stack state that are the enrichment of the state with prioritized variants. And these are prioritized variants that are ranked by 14 different methods that's been published before to use to prioritize the different genetic nucleotides based on how conserved they are, or how likely they are to have an effect on phenotype, etc. And you can see that 7 different scores show that the top 5 most enriched states are exclusively in a promoter or transcription stat size state. PHASTCONS score, for example, show a different patterns. In this case, PHASTCONS actually show depletions in promoter state, and the reason is we state the reason. It could be that PHASTCONS actually prioritize bases of the genome that are depleted in human genetic polymorphism. However, these region associated with these states tends to show increased enrichment at CpG islands, and therefore show increased mutation rates, and therefore they tend to be depleted with regions that are prioritized by PHASTCONS. And there's also other association addressed in our manuscript. And the point of this particular analysis is that different score tends to prioritize bases that are in different epigenomic contexts of the genome. And this analysis is not really made to show that one score is better than the other. We were just trying to highlight the differences in epigenomic context or chromatin state context that different scores tend to highlight. Lastly, we look at the full stack annotation state enrichment with cancer-associated somatic mutations. And here I show you the top state that are enriched with somatic mutations found in different types of cancer. And you can see that most of these states are again associated with the H3K9ME3 mark. They are either heterochromatin state or usually heterochromatins or quiescent state, and we also show some state in the group of gap, assembly gap, and artifact state. And we actually looked into many different papers, and we found out actually around 3 to 4 papers that are associated, that show the associations of H3K9ME3 with cancer associated somatic mutations. And one explanation for this patterns is that in regions that are associated with H3K9ME3 heterochromatic region, they tend to, because they are packed in heterochromatin, they can have less access to DNA mismatch repair mechanism. And the gap and artifact state enrichment right here may be because of false variance call. So in conclusion, we actually applied the stack modeling approach of CHROMHMM to more than 1,000 data sets that provide 32 chromatin marks in 120 different cell types. We show that and we call this the full stack annotation. The full stack annotation actually shows some state with constitutive versus cell type specific chromatin signals. We conducted many analysis to characterize the state's biological implications. We also characterize the state enrichment with different conserved elements, repeats element, different types of genetic variations, etc. You can also download our full stack annotations and use the characterizations of the state that we provide for your own analysis to understand the epigenomic context of different regions of the genome that are of your interest. Actually, I can show that this is some of the this is just to show you the additional files that come with our manuscript that you can use to understand the characteristics of the different state. And it's actually, we compile into really comprehensive sets of additional files for your reference. Sorry. So the full stack annotations that we generated has been used in our collaborators' studies to study the context of methylations and maximum lifespans across different species. And we are also trying to finalize another full stack annotations, not in the human, but this time in the mouse model. And with that, I'd like to acknowledge my professor, Jason Ernst, and members of the Ernst Lab and our collaborator for meaningful and helpful feedbacks, and my funding, and QCBio for giving me this chance to present. Thank you, and I'm happy to take any questions.